Hi Year 11, I'm really sorry I couldn't be there in person. I'm sorry I missed your lesson this morning. It's not ideal timing, but hopefully this video is going to be the next best thing. So just follow through with the paper that Sir's handed out for you. If you've got any questions, please ask him. Pause the video, have a go at the questions yourself, and work through it with me, please. We're probably going to run out of time, so I'm not going to be able to do the whole paper on this video, but I have included the mark scheme, so please use that after the session. Best of luck. See you tomorrow. Okay, so let's have a look at question 1. So here we have a question on microscopy and magnification. So the image below shows some muscle cells from the wall of the stomach as seen through a light microscope. We get some pictures of some muscle cells from the wall of the stomach, and we've got a little bar showing that this distance in the image is represents 0.1 millimeters. The first thing it asks us to do is describe the function of muscle cells in the wall of the stomach. So the job of the stomach really is to churn the food around. So these muscle cells contract, which means they shorten to move, churn the stomach. Okay, so if we have a look at the next question, we've got this. So the figure above is highly magnified. The scale bar in the figure above represents 0.1 millimeters. So use a ruler to measure the length of the scale bar and then calculate the magnification of the figure above. So magnification is equal to the size of the image divided by the actual size of the object. So we need to work out how big the image is, and then we're actually given this, which helps us massively. So we don't need to actually measure these cells because we've got this little scale bar there. So we'll take our ruler and place it here, and that appears to me like we're on, I would say, 39 millimeters. We need to use the same units for our image size as the object size, and they've used millimeters here. So we have 39 divided by 0.1. We'll take our calculator, and that gives us 390. And as always, make sure you write your answer here. Now, when you've got a question like this, there will be a little bit of leeway either way. They might accept 400 or a little bit less than 390, because there's always a small amount of error when taking a measurement. Next, we've got muscle cells in the figure above contain many mitochondria, so you can see those here. So the function of mitochondria is to transfer energy via respiration. So if it had been a single mark, we could have just said it's where respiration takes place. However, for 2, we need to say what the point of respiration is. So we're transferring energy via respiration. Next, the muscle cells above also contain many ribosomes. The ribosomes cannot be seen in the figure above. What is the function of a ribosome? So it's pretty straightforward. It is for protein synthesis, and we've spoken about this quite a lot in the last few weeks. So it's where your cells make any proteins they need. Suggest why ribosomes cannot be seen through a light microscope. They're simply too small. So you can't magnify enough with a light microscope to be able to make them visible. What you'd have to use is an electron microscope. Okay, so let's take a look at question 2. The image below shows part of a root from a cress plant, and we've got this image in front of us here, and it's quite a strange image. It looks almost fake, but this is the sort of image you'd get from an electron microscope. This bit here I think is a little bit misleading. If you'd look, if it tells us it's got a magnification of times 200, that's normally the sort of magnification you'd see with a light microscope. However, you can tell just from the image this is an electron microscope. So what type of microscope was used? Electron microscope. So here we've got the magnification of the crest root in the image above is times 200. There are 1000 micrometers in a millimeter, and we need to calculate the real length of the root hair X, giving our answers in micrometers. So what we're going to need to do is convert units. We're going to use the same equation as we did previously. Look, we're going to use magnification equals image size over object size. So I'm going to write that here. So m equals I over O. And next, just as we did before, we're going to have to take our ruler and we're going to have to make a measurement of this cell structure here. And that looks to me like it's 30 millimeters, 3 centimeters. So my image is 30 millimeters. It's 30 millimeters in length. And again, I need to do a conversion now. I need to go into micrometers, and it tells us there are 1000 micrometers in a millimeter. So I can do a little conversion thing here. So if I want to go from micrometers to millimeters, I know that 1 millimeter is 1000 micrometers. So if I want to go from these to these, I need to multiply by 1000. If I wanted to go the other way, I'd have to divide by 1000. So I'm going to take my 30 millimeters, I'm going to multiply it by 1000 because I want to go from millimeters to micrometers. So 30 multiplied by 1000 gives me 30,000 micrometers. So this is still the size of the image. I haven't even looked over here yet, so I've just been doing my conversions. So I'm looking for the real length of the root hair. So that's the size of the object. So I can rearrange that by putting O on the top and moving M underneath. So image size divided by magnification. So 30,000 over 200 gives me a 150 micrometers. And just so the examiner knows what they're marking, I'll put a nice big underline there. Oh, I missed off the actual little box for the answer, never mind. Root hair cells take up water from the soil. Explain one way in which the root hair cell is adapted to this function. So I think the key one that you always need to mention would be they are long and thin, which gives them a large surface area to volume ratio. So they're long and thin gives them a large surface area to volume ratio. The fact that they're thin also gives them a short diffusion pathway, so they don't have to move very long. So next, the table shows the water uptake by a plant's root on 2 different days. Explain why the mean rate of water uptake is higher on a hot day than on a cold day. So this happens via the process of transpiration. So water moves through the plant via transpiration. So water comes into the root hair cells, moves up the xylem structures in the plant through the leaves, and then it evaporates out of the stomata. On a hotter day, with a higher temperature, you have a higher rate of evaporation, which gives you a higher rate of transpiration and hence a greater water uptake. So a higher temperature means more evaporation, hence higher water uptake. The concentration of mineral ions in the soil is lower than in the root hair cells. So if you imagine you've got your root hair cell here like that, you've got lots of mineral ions inside, but not many outside. However, your cell still needs to move those mineral ions inside it so it can use them. So we are going against the concentration gradient here, so we know that this is going to be active transport. So we can say the mineral ions move against the concentration gradients. So we know that this must be active transport. And as always, we remember EROM. So this needs energy. Which it gets via respiration. It needs oxygen. And this process takes place in the mitochondria. So we've talked about we need energy. We've talked about how we get that energy. It's provided by respiration. We've mentioned oxygen. It wasn't strictly necessary in this question, but it's good for completeness. And then we've said that this takes place in the mitochondria. So for lots of AT, for lots of active transport, many mitochondria are required. Okay. And that's it for question 2. Okay, so let's have a look at question 3. This one is about malaria. So it starts off with having to describe what happens during the liver infection stage of the life cycle of the malaria parasite. So your mosquito, which carries the spores, the sporozoites of the malaria parasite in its saliva, bites, and those sporozoites end up in the liver, in the liver cells. So in the liver cells, those are sporozoites from the mosquito saliva reproduce, and you end up with many thousands lots and lots of these, of things called merozoites, so the next stage in their life cycle. So starts off as spores, sporozoites, and they end up as merozoites. And once they've reproduced, once they've matured, these are released from the liver into the blood cells or the bloodstream, and that's where they cause their damage. The next question looks at some information about the development of a vaccine against malaria. So what have we got? We've got scientists have removed 2 important genes in a malaria parasite. The malaria parasite causes the type of malaria most deadly to humans, so we obviously want to get rid of that. When the genes are removed, the malaria parasite stays in the liver infection phase, so they stay as those merozoites and they don't get released, so stopping it spreading to the bloodstream where they cause the disease and they cause their actual harm. Okay, so that's good. We want to stop that happening. Scientists are using the genetically modified malaria parasites to develop a vaccine against malaria. Similar vaccines have been tested in mice, that's interesting, and produce a 100% protection against malarial infection. Okay, that sounds good as well. Scientists hope that the vaccine will produce similar results in humans. Okay, so we hope that it will produce similar results in humans, so we don't know that yet. Although 2 genes have been removed, the parasite is alive and able to stimulate the body's protective immune system. So it triggers your immune system into a response, and it makes phagocytes and lymphocytes that they're able to attack these parasites. So it destroys them once they've come into the body. Scientists think that the weakened parasites used in the vaccine will not become harmful again because the genes have been removed from the genetic material and they cannot recreate the gene. Okay, so we need to use this information and evaluate the use of this vaccine. So we're going to need to look at some pros, some cons, and then decide what we think. So I'll talk about pros first. So the good points. So it reduces the incidence of the disease. Which, you know, we're getting fewer cases of the disease, so that's good. Success because it works in mice, it will probably work in humans. You know, that's why we use mice as an analogy for humans. And what else? So it prevents the release of merozoites into the blood. So that's what we want to stop. That's what causes us to get ill. On the negative side, well, it works in mice, but that doesn't guarantee it's going to work in humans. So one con would be success in mice doesn't mean necessarily, it probably does, but we don't know. We'd have to check for side effects. What else? So could cause possible liver damage. We're still getting the sporozoites into the liver and they're still reproducing in there, so it could cause liver damage. Anything else? We're still injecting a living parasite as well, so that's probably not the best. So still injecting living parasites. So possible risks. So you've got a possible risk there. I think that's it for question 3. So question 4 is another question about how to reduce infections. So we've got we're looking at lungworm, which is something that affects dogs. It's caused by a small worm. And we've got the life cycle here. So let's see what we've got. Lungworm. Lungworm reproduces in the dog and infects the lungs. The larvae, which are the young stages of the worm, are coughed up from the lungs and swallowed. The worm larvae pass out in the feces, so the dog mess, the poo, and are picked up by snails. And then other dogs come along, see those snails, they touch them or they eat them, and that passes on the disease to the next dog. So what type of organism is represented by the snail in the life cycle of the worm? So we need to remember that if it's a living thing that transmits disease, it's a vector. If it's non-living, then we call that a vehicle. So for example, when we spoke about malaria a second ago, the mosquito is a living thing, so the mosquito is the vector. If we're talking about tetanus, so if you step on a rusty nail and get tetanus from that, then the rusty nail is the vehicle that infected you. So let's have a look at our options. Fungus, parasite, protist. Well, it's none of these three, so it's got to be that vector. Next, suggest how the spread of the lungworm disease can be prevented. So we've got a few options. As always, we can get rid of the vector. So we can destroy those snails. Probably not the best. If we do notice that any of the animals are infected, if we isolate them, then their dog mess isn't going to get eaten by snails, and then it's definitely not going to get touched by other dogs. We could treat the dogs as soon as they're sick, and that stops them passing on the parasites. And to be honest, if you're a dog owner, you need to be picking up your dog mess. So we need to educate owners about picking up dog mess. So that's why I had to squeeze it in there. There was lots of space on the next page. So next disease, malaria is a disease spread by mosquitoes. Again, we've spoken about this a lot. 2 ways to control the spread of malaria. So again, we need to reduce the number of vectors. We need to get rid of the number of mosquitoes. So we could stop mosquitoes breeding, would be an easy one. So they breed in like stagnant ponds. So to do this, we could remove standing water or stagnant water. So rivers they're not able to breed in, so rivers are fine, it's just small ponds and lakes. We could also use mosquito nets. That's a very, very good one. So those are nets that you put over your bed when you sleep at night, and the mosquitoes can't get to you to bite you. So the fewer bites you get, the less likely you are to get malaria. Also, you've got the idea of insect repellent, so you can put some DEET on yourself to stop yourself getting bitten. Question 5 looks at the blood and diseases. So what have we got here? The parts of the blood can be separated by spinning the blood in a centrifuge. So that's just a machine that spins things round in circles. The heavier things get pushed to the outside, and the less dense substances kind of stay towards the middle. So you end up with something that looks like this. So your blood is separated into its component parts. We've got 10 centimetres cubed of it, and we've got the plasma at the top here. That kind of makes up 5.5 centimetres cubed. A tiny amount is your white blood cells and your platelets. So your white blood cells, they're part of the body's defence. Your platelets help the blood clot, and the plasma carries dissolved substances, and the red blood cells carry oxygen and carbon dioxide. It's asking us to calculate the percentage of blood that is made up of plasma. So as we just mentioned, we've got 5.5 centimeters cubed. So 5.5 centimeters cubed is the plasma. The total amount is 10. So 5.5 over 10, and because it's a percentage, we times it by 100. 55%. On to our next bit. 3 chemical substances transported by plasma, and there's absolutely tons that we could look at. The most common, I think, are urea. So your byproducts of proteins. You could have glucose. You can have proteins themselves. You can have amino acids, antibodies, antitoxins. You know, the list is very, very long. Next, we've got an extended writing question. How nice. And we've got white blood cells are part of the immune system. White blood cells help the body to defend itself against pathogens. Describe how pathogens cause infections and describe how the immune system defends against this. Okay, so let's start off with the idea of pathogens. So we're looking at bacteria, really, and viruses are these pathogens. They reproduce inside the body, and they do that rapidly, so they do it quickly. And bacteria, when they're reproducing, when they're doing their stuff, they produce poisons. They produce toxins is the word that we use, and it's these toxins that make us feel sick. Viruses reproduce inside cells, and that causes damage. So it destroys the cell, basically. So that's how they make us sick. So how can we defend ourselves? So we have white blood cells. They can ingest or engulf pathogens. So basically they eat them. They also produce antibodies that destroy specific pathogens. So the white blood cell themselves will try and engulf anything they find. These antibodies are targeted towards specific pathogens, specific diseases. So pathogens would be better than diseases there. And they also produce, so the white blood cells produce antitoxins that deal with those poisons, those toxins that were produced by the bacteria in the first place. And this process leads to immunity from that pathogen. So a harmful microorganism, a pathogen, comes into your body. The first line of defence is that the white blood cells will try and engulf it, ingest it. They will also produce antibodies and antitoxins that are kind of specific to that pathogen. And once your body's produced these antibodies, it remembers how to make them. So if it's ever reinfected with the same pathogen, then it's already got those antibodies ready to go to attack the pathogen, and you don't get sick in the first place. So question 6 looks at the experiment we spoke about the day before yesterday, I think. So we've got light intensity, carbon dioxide concentration and temperature are factors that affect the rate of photosynthesis, and we need to know how we're going to investigate the effect of light intensity on this rate of photosynthesis, and the image below shows you some of the apparatus you might use. When I say below, I mean on the next page. So we have a lamp, we have some pondweed, we have a ruler, and we have a beaker, and apparently these are not to scale, so yeah, it's not a giant beaker. We need to talk about how we're going to set up the apparatus, what measurements we're going to take, and how we could make sure it's a fair test. So we're talking about, is it valid? Do we keep control variables constant? So we're going to change one thing, which is going to be the light intensity. We're going to measure another, which is the rate of photosynthesis. Everything else needs to be kept the same, otherwise it's not valid, so it's obviously not a fair test. So what are we going to do? We're going to put the pondweed in the beaker. With some water. We will also use something called sodium hydrogen carbonate. If I can spell it. And the job of this is to provide carbon dioxide, and we'll keep this concentration of sodium hydrogen carbonate constant to make sure it's valid, given as a fair test. So this allows us to control the concentration of carbon dioxide, which we need to keep the same. So put the pondweed in the beaker with the water and sodium hydrogen carbonate. Place the lamp. Let's start off with 5 centimeters from the beaker. And we would count the number of bubbles of oxygen. We know it's oxygen because photosynthesis produces oxygen. So count the number of bubbles of O2 produced each in 1 minute. This will give us it's an indication of the rate of photosynthesis. So this is our dependent variable. This is what we're measuring. So that's our first reading. So we can vary the light intensity by moving the pondweed further away from our light source. So the further you are away from the light source, the lower the light intensity will be. So we could do 10, 15, 20, 25, 30, 35, 40 centimetres. So we've got a good range of results there. It's probably more than we need. 7 would be the minimum. If we count 5, we've got 8 there. So that would be good. And count number of bubbles. Per minute each time. So we've talked about that. We need to talk a little bit about these control variables and how we might be able to make it a little bit better. So we're probably going to repeat this a further 2 times, take a mean average. It hasn't actually asked us about reliability in the question, but by repeating it makes it easier to spot any errors, and we can discard any anomalies, so that increases the reliability of the investigation. Doesn't affect validity though. We could control the temperature, because that's a factor that affects photosynthesis, by using an LED light that doesn't give out as much heat. They're more efficient than filament lamps. And we could use same length of pondweed. If we use more pondweed, we're going to get more bubbles. That's pretty obvious. We could also talk about, well, if we're counting bubbles, that's not super accurate. You know, bubbles could be of different sizes. So we could also use a gas syringe to measure the volume of emitted oxygen rather than counting bubbles. So that would make it a bit better. So yeah, that should get us what we need. A diagram would have been nice, but I didn't have space. So we've got question 7 here. This looks at how substances are transported through plants, and the main part of this is going to be how water is taken up through the plant via the process of transpiration travelling through the xylem. So let's have a look at what we've got. We've got water is transported from the roots to the stem of the plant in the xylem. So it's the xylem tissue that transports water. Dissolved sugars, so your glucose, are transported through the phloem. So xylem is water, phloem carries sugars. The movement of water through that plant, as I mentioned a second ago, that's transpiration. And water vapour moves out of the plant through pores, and we spoke about this a little bit earlier in the week. They've got those guard cells surrounding them. They're called stomata. Next, what have we got? We've got an investigation into the effect of different conditions on water loss from the leaves. So we've got some water in a conical flask. We've got a plant put through there. We've got some cotton wool surrounding the stem of the plant. That's probably to stop water evaporating or escaping from the conical flask via evaporation. That means we know that any water that is lost is via transpiration, so it stops that evaporation happening. And we've got it on a balance, so we can measure the difference in mass. So we'll look at the mass at the start of the investigation and at the end, and that difference will be the amount of water that's taken up via transpiration. So they did 4 flasks, A, B, C and D. They used the same size plant shoot in each flask, recorded the mass of the flask and plant shoot at the start of each experiment, put them in different conditions, and then they recorded the mass of each flask and plant shoot after 2 hours. Okay, so these are the different situations they had. They had 2 at 20 degrees Celsius, and one had a fan blowing over it and one didn't. They did it at a higher temperature, 35 degrees Celsius, and again one had a fan, one didn't. So you can compare these two, because you can look at the temperature and whether or not they had a fan, and you can compare, for example, these two or these two, because they had similar conditions. They had no fans, but the temperature was changed. So why did we use the cotton wool in each flask? Oh, well, we mentioned that a second ago, didn't we? We did that to prevent any evaporation of water from the flask. So we're not interested in evaporation from the flask, only in uptake through the plant. From the flask. We used the same size of plant shoots in each of the investigations, and we did this to have the same surface area of leaves. Sorry. And then from which the same amount of water would evaporate. So, you know, if you had one with loads and sorry, I can't even see that. If you had one with loads and loads of leaves and one with only 2 leaves, obviously if you've got more leaves, you've got a bigger surface area, so you're definitely going to have lots and lots of uptake if you've got lots and lots of leaves. Apologies for not showing you that. So same surface area, lots of mistakes there, sorry. Same surface area of leaves from which the same amount of water would evaporate. Next, we've got a bit of a confusing table here. We've got the results, but with my copying, I've cleverly split it over 2 pages. So you've got the results for A here. So that was 20 degrees, no fan. Started off at 150, ended up at 148.1, and it's lost 1.9 grams of water over those 2 hours. If we flip over to the next page, we've got the rest of the data. So annoyingly split, sorry about that. It's asking us about D, so what's the mass of water loss? So we'd have to do the final, the initial minus the final. So we had 150.0. At the end we had 145.5, and the difference between those 2 numbers is 4.5 grams. So 4.5 grams. Not too bad. And I'm going to pop it into the table up here as well, just in case I need to do a comparison a little bit. Suggest what conclusion can be made about the effect of temperature on water loss from the plant shoot. Okay, so we're going to compare B and D. And no, we're not. That's a lie. We're going to compare C and A. And the reason for that is because neither of them had a fan, but the A had a temperature of 20 degrees and C had a temperature of 35 degrees. So when the temperature was higher, it lost 3.1 grams, whereas when it was colder, it only lost 1.9 grams. So that's significantly less. So we can say the higher the temperature, the higher the rate of water loss. So the higher the temperature, the higher the rate of water loss, so evaporation from the leaves. What conclusion can be made about the effect of the fan on the water loss from the plant shoot? So what effect does the fan have basically? So let's have a look. Let's use C and D for an example. So these are both at 35 degrees. Without the fan, we lost 3.1 grams. With the fan, we lost 4.5. So again, that's almost 50% more than with the fan. So having a lot of air, so moving air, increases the rate of water loss. And that's because you're always you're maintaining a high concentration gradient. So you've got or you've always got sort of dry air outside the leaf if you're moving the air away. Whereas if you don't have the fan, then the air by the leaf becomes humid. It contains lots of water, so evaporation doesn't happen as efficiently. They carried out another experiment, 20 degrees with no fan, and they used this equipment here. So this kind of links to the idea we just spoke about. As well as having the cotton wool, they've got a plastic bag sealing it. So any water vapour that does leave the leaves is trapped in here. So this bag, I would imagine, will get incredibly humid, very, very humid. Record the mass of the plant. So they did the same. Yep, they removed the bag after 2 hours, recorded the mass again. What mass of water will be lost from the shoot? And I think it's because it's got this plastic bag around you, you're not going to lose very much at all because you have almost no airflow. So I think it's got to be this 0.3 grams, the smallest one there is, and that's because the plastic bag reduced airflow across the leaves. We could talk about humidity gradients. It gives it a very low humidity gradient because you've got humidity right around the leaves, so you're going to have less evaporation. Okay, year 11, we're going to leave it there. I will see you tomorrow. Thanks very much.